Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. This is the second lecture of Neurobiology series where we are going to discuss about neurons, the nerve cells, and we will have an overview about our nervous system. In the previous lecture, we learned about the history of, history of neuroscience. We learned about how different researchers came up with the different observation and they concluded what our nervous system is made up of. So we discussed about doctrine by Santiago Ramanujan, as well as we discussed about the reticular theory, which was discarded but given by Camillo Golgi, and how both of them showed they contributed to the field of neuroscience. So today's lecture we will be learning about neurons. So before learning about neurons, let's have a quick overview of our nervous system. How does our nervous system works? What it is made up of? Okay, so our nervous system, the human nervous system, it is divided into two major subdivisions. The first one is known as central nervous system, and the other is known as peripheral nervous system. The central nervous system, as the name states, is central. So it is central to all of the information processing, all of the information receiving. So this is central part of nervous system, which has two main components: the brain and the spinal cord. So we know that brain is kind of like a headquarters for all of the information. So brain does the job. Brain does all of the job. Like it receives the information from the different body parts, and then it processes that information, and then it gives out the signals to the specific muscles or glands upon how to work. Like brain is the central part. Brain is central to all of our behavior. So that is why it is comprised in the central nervous system. So we already know about brain and spinal cord, and they make the central nervous system. So the another subdivision is peripheral nervous system, which is comprised of the all of the nerves which arise from brain and spinal cord. So the nerves which arise from brain, the cranial nerves, and the nerves means the neurons that arise from spinal cord. They are known as spinal nerves. So cranial nerves, spinal nerves, and ganglia, all of them, they make up the peripheral nervous system. So the nervous system, it is divided into two parts: central nervous system and peripheral nervous system. The central nervous system has brain and spinal cord, while the peripheral nervous system has cranial nerves, spinal nerves, and the ganglia. Okay, peripheral nervous system is again divided into two parts, which we depending upon how they act: the somatic nervous system and the autonomous nervous system. Okay, the somatic nervous system controls all of our voluntary actions, like when the brain gives out a signal to a skeletal muscle, and you start walking. So it's like all of the voluntary action, they are carried out by somatic nervous system, the somatic peripheral nervous system, while the autonomous nervous system carry out all of the involuntary functions. So somatic carry out the voluntary functions, and the autonomous nervous system carry out the involuntary function. Okay, so involuntary functions like your beating of heart, and then digestion, all of the involuntary activities, they are done by the autonomous nervous system. The peripheral nervous system, it is a connecting thing between the central nervous system and our body. So like whatever stimulus your body is receiving, that is conducted by peripheral nervous system to the central nervous system, and then when that signal goes to the central nervous system, that information is processed, and upon that, whatever your behavior is going to be, that signal is then transmitted from the brain to the nerves, to the peripheral nervous system. So so peripheral nervous system is kind of the connecting link, the link between the central nervous system and your body. So this somatic nervous system controls the voluntary action, while autonomous controls the involuntary actions. The autonomous nervous system, it is again divided into two parts. Okay, two parts. The first one is parasympathetic nervous system, and the other one is sympathetic nervous system. Okay, so this division, so this categorization is done, it is based on, like, on during which condition your peripheral nervous system is acting, your involuntary peripheral nervous system is acting. When you are in, you're in at rest condition, you're calm, you're doing your daily routine, you're following your daily routine, you're doing regular things that you usually do. So when you are at the state of rest, the nervous system, which the autonomous nervous system which is working at that point of time, is parasympathetic nervous system. Or on the contrary, when you have the fight or flight situation, like you're in some danger, or you're nervous, or you're feeling some excitation, at that point of time, in the fight or flight condition, the sympathetic nervous system is working. Okay, so sympathetic nervous system takes control in the fight or flight condition. Okay, so this was a quick overview about our nervous system. I hope it is clear to you all. So our nervous system, like it's a system, right? So system, it is made up of so this system is made up of nervous tissue, and this tissue will be made up of a lot of cells, right? So let's learn about which type of cells the nervous system is made up of. So nervous system is made up of two main types of cells. The first one, which we are going to cover in this lecture, are nerve cells, which we also call as neurons. Okay, and the other type of cells is known as glial cells. Okay, so the nerve cells are very important ones, and we already know that they are the ones which are going to, produce the signal and conduct the signal. So nerve cells, what happens is that whenever there is some stimulus, that stimulus is, that is received as a signal, and that signal is then conducted to the consecutive nerve cell, or it can be conducted to the effector muscle or effector gland cell. So whatever the signaling part is, that signaling part is the job of nerve cells, and that is why the nerve cells are also known as signaling units of the nervous system. So the nerve cells are known as signaling units of nervous system, because they are going to carry out the signaling part. So whatever nerve impulses conduction is done, that is seen only in nerve cells, and that is why nerve cells are the cells, they are the excitable cells. Okay, they're the excitable cells. So they can be excited, so means whenever there is a stimulus, so they get excited as a response to that stimulus, and that is why the nerve cells are also known as signaling units. Okay, while on the contrary, the glial cells, they are not excitable, though they are much more similar to nerve cells. Their composition is similar to the nerve cells, okay, developmentally and anatomically, but yeah, there are structural differences that we will be covering in the lecture specific for glial cells. But the glial cells, they are unlike the nerve cells, they are non-excitable, so they can't be excited, so they can't respond to a stimuli. Okay, so they are not excitable, so they are non-excitable cells. And glial cells are they are very important, because the majority of nervous system is made up of glial cells. So we all know about neurons, but besides neurons, there are glial cells which are very helpful, because they are going to support the nerve cells. So they are going to carry out a lot of important functions, like supporting the nerve cells, protecting the nerve cells, and providing nutrition to nerve cells. They will act as immune cells of our nervous system, the microglia and all. So we will be having a separate lecture about glial cell. In today's lecture, we are going to we are going to learn about neurons. Okay, so here I have listed down the topics that one by one we are going to cover in upcoming lectures. So the next 5 to 6 lectures, we will be covering those topics. The very first thing that we are going to learn and discuss is about the structural components of nervous system, specifically nerve cell. So this lecture is dedicated to neurons. So in this lecture, we are only going to learn about the structural components of neurons. So like what is the structure of a neuron, and what the specific part of neuron does. so we are going to also discuss about the jobs of that parts. Okay, so we are going to only discuss the structural properties of the neurons in this lecture. Then we will also cover the mechanism by which nerve cell produce signals. So we will learn about how upon receiving a stimulus that the neuron, it is able to release the neurotransmitter, and how means by what are the properties of those neurons that they are able to produce signals unlike the other cells. So we will be discussing all of those, and by what is the procedure by which they produce signal and they conduct signal. So we will also learn about the mechanism part. Okay, so we will be learning about that in separate lecture. Then we will learn about the patterns of connections between the nerve cells. So I hope you remember, if you have not watched that video about the history of the his history part of the neuroscience, where we discussed about the observations done by Golgi and Ramanujan. So both Golgi and Kyle, both of them observed a thing, the reticular theory and the neuron doctrine given by Kyle. Both of them had one thing in common, and that was that the neurons, the nervous system, it has a connection of the cells. Okay, but Golgi said that they form the nerve cells, they form a continuous network. So did not say that they are individual cell. Rather, he said that all of the cells, they form a continuous network, and then they have a cumulative effect of whatever stimulus is going to be given to that system. On the contrary, Ramanujan said that it is not a continuous syncytium, rather it is the network of the discrete cell. So he stressed on the individuality and the discreteness of each cell. So he said that each cell is a discrete cell, and it has its own individuality, it has its own identity, right? But both of them did observe a connection. So we are going to learn about the, so we know that those connections, they are not random, but they actually have some patterns. So we are going to learn about the patterns of the connections between the nerve cells, like how the nerve cell forms the synapse with the consecutive nerve cell. So we are going to learn about those patterns of connections between the nerve cells, as well as between nerve cell and its target. So those connections are not just between two nerve cells, but they are also in between the nerve cell and its target. Like for example, a single let's say a neuron, it is controlling some skeletal muscle. Okay, so it is going to form a connection with that particular muscle as well, so that they are known as neuromuscular junctions. So such connections, we will also learn about such connections, the pattern of those connection. Or let's say that there is a neuron, and that is forming a connection with an gland. So the connection between the neurons and the effector gland. So we are going to learn about all of the patterns of those connection between nerve cells, as well as between nerve cells and their target their target bodies. Okay, their targets. So we will have a separate lecture on this, and then we will cover the relationship of the different patterns of interconnection to the behavior. So how those different patterns, they are they are able to result into certain behavior. For example, we will learn about some reflex action in this lecture, how the different neural circuits, they are going to result into certain behavior, like how a certain kind of wiring is going to result into you acting in a specific manner. So we will be covering that in this lecture. Then we will also discuss about how the neurons, how the neuronal connection, they are modified by behavior. So whatever they are not static, they can be modified, and that we will also discover in the last lecture of this series. So this is the basic overview that we are going to discuss, that we are going to have for next 5 to 6 lecture. I hope it's clear to you all. So now let's move on to the neurons. Let's learn about neurons. Okay, so this is a picture that I've taken from Wikipedia, so it's easily available, it will be easily available to you all. So here you can see a neuron. Okay, so nerve cell. Okay, so let's just discuss about cells. So whenever you say cell, what comes into your mind? There will be a certain compartment with plasma membrane, right? There will be a plasma membrane. Okay, and in which there will be cytosol and a lot of organelles, let's say some mitochondria. It will have lysosomes. Okay, obviously the central the headquarters, nucleus, How could we forget about nucleus? Okay, so the cell will have, plasma membrane, it will have nucleus, it will have a lot of organelles, mitochondria, lysosome, and then, the extension of nucleus, the the Golgi bodies, so on and so forth. So whenever we say cell, this kind of schematic comes into your mind, okay? But nerve cells, they are somewhat different. So they do not have only this thing, but they have some processes. So the main important body, which it is similar to all of cells, it is almost similar to all of the cells, but it has one more thing, that is processes. So there will be some extension of plasma membrane from this cell body that we call as cellular processes, and that is why neurons are quite, they're quite interesting cells, because they have a complex structure, and which is quite distinct and which is quite different from normal cell. So here you can see there will be a centrally located cell body, which is almost similar to all cells. It will be having its own, plasma membrane, it will be having its own nucleus, and then the organelles. Here you can see it's here you can see this cell body. It has the, nucleus, it has endoplasmic reticulum, it even has Golgi apparatus, the mitochondria. So it has all of the cell organelles. So the cell body is the central part of the neuron, okay? So the cell body is central part of the neuron, which is going to carry out all of the metabolic functions, okay? It is also known as soma. So this is cell body or soma. Okay, let me write it in a different color. So it is cell body or soma. Okay, so the cell body, it is the metabolic center of cell. So all of the metabolic pathways, all of the, no, any cell needs, protein in protein synthesizing machinery, because the cell will, central dogma of life. Remember that from the DNA, you are going to, DNA is going to be transcribed to RNA, and then it will be translated to protein. So nerve cells, they are also cells, so they need to synthesize protein and all. So all of those metabolic pathways, they even need energy. So all of those metabolic pathways, that will be done by the cell body, and it also has nucleus. So all of the genes that will be, comprised in the nucleus, and so the nucleus will be the controlling center of that neuron, and that resides in the cell body. So cell body, just like any other cell, it works and it functions, okay? But the different part comes here. This cell body has two different type of processes. The tree like branching process, which is known as dendrite, and the second one is, long and tubular process, which is known as axon. So let me write it here separately, that cell body, it has two processes. The tree like branching ones, they are known as dendrites, and the tubular and a long one, it is known as axon. So both of the, remember, both of them, they arise from cell body, okay? In this case, in this specific case, when we learn about the types of neurons, you will see from where they arise. Anyways, so from cell body, there will be two types of processes, axon and dendrites. So here you can see the dendrites are tree like branching multiple, so they are short appendages, and they have multiple branches, okay? So it is like tree like branching pattern. So these are known as dendrites. And then there will be a single long tubular process which also arises from cell body, which we call as axon. So let's learn about dendrites first. So dendrites are, okay, I hope you remember that when we were discussing about Kyle, so he gave his, in the neuron doctrine, he said that those are the connection of discrete cell, but there was a second point that he also mentioned, and he said that the neurons have polarity. Okay, so let me explain it a bit. Okay, so the neurons, they are, ectodermal in origin. So just like epithelial cells, they also have the polarity. So epithelial cells, they have apical basal polarity, right? So there will be a basal part and there will be a apical part. In the same way, neurons also have polarity, and we will discuss that in upcoming lecture. Okay, let's discuss it here. So this polarity, this is this is based on the conduction of the nerve impulse. Okay, so this is based on the conduction of the information, the relay of the information. So the way apical basal polarity is seen in epithelial cells. The same way in nervous cells as well, there is apical basal polarity. So the basal part in the epithelial cell, it has the nucleus part. So this basal part, it has nucleus. So this is the basal part of the neuron. Well, this is known as apical part of the neuron, which has the, most of the cytoskeletal elements, just like epithelial cells. But the polarity, basically, whenever you mention polarity in case of nerve cells, in case of neurons, it always states that information will be relayed from one side to other side, and that is why it is known as, this is because they are polar in nature, so they have polarity. So the dendritic part is the part where you are going to receive the input. Okay, so the polarity will be like, the flow of information will be like from dendrite, okay, to the axon hillock, then to the axon, and then to the synaptic terminals. Okay, so this will the information, so stimulus will be received here, and then it will be passed down like this to the next neuron. So this, let's say this is the next neuron. Okay, so this is how the polarity is seen in the neurons. So what happens is that the dendritic part is the part where the this is the input part. So this is going to receive the input. So whatever this branching pattern you can see here, it is going to receive the stimulus, and then the signal will be generated here, and that will be passed down. Okay, so this whatever signal is generated at dendrites at the this side, it is known as local signal. Okay, so if it crosses certain threshold level, it is relayed to the axon hillock. Okay, so axon hillock is also known as trigger zone. It is a very important zone, because if on the trigger zone, that the value, the amplitude of that signal, which is already generated in dendrite, that amplitude, if it crosses certain threshold level, and suddenly if it crosses, two things are important. First, it should cross threshold, and second, it should the crossing of threshold should be sudden. Okay, so when this happens, the action potential will be generated. Yeah, we will discuss about action potential and resting membrane potential. Do not stress about it too much. Just, in layman terms, let's say that the when there will be a signal. Okay, when, let's say that I pinched you, and this is the sensory neuron, so this will be sensed by the dendrites, let's say. So there will be a generation of local signal. Okay, once this local signal is generated in dendrite, once this crosses the threshold, that will be once this crosses the threshold, that will be passed down to the axon hillock. And axon hillock is known as trigger zone, because at this point, all of the stimulus, that, stimulus could be multiple. So they are not, there will not be a single stimulus. So all of them, this multiple graded stimulus, that they will be, integrated in the axon hillock part. And once when they are integrated in axon hillock, that creates an action potential. Do not, if you don't know about action potential, do not fret. Just remember that the action potential generation site is the axon hillock. Okay, so at the axon hillock, the action potential is generated. So it's like a signal which is going to relayed throughout the neuron, and this relaying of the nerve impulse, that will be done in the axonal part. So this axon, so at the trigger zone, action potential is generated, and in the throughout the axon, this action potential is passed on. Okay, so this action potential travels through the axon all the way to the end parts, which we known as which we call as, the end parts are known as telodendria. So this axon also have some branching pattern, and at its end, which is known as telodendria, which has, the ends of which are known as synaptic terminals, because this is going to form a junction with the future the consecutive neuron, and that junction is known as synapse. Okay, and that is why those are known as synaptic terminals. So this is the output part of that single neuron. So the input part, the input of information will be done from dendrite, then the signal generation will be done at axon hillock, the action potential generation, and then that action potential will be passed down throughout the axon, and at the synaptic terminals, this is kind of the output for this is the output of that neuron, and that will serve as an input for the next neuron. So this so this first neuron, okay, so this is the synapse, right? So this is the first neuron, and this is the second neuron. Okay, let me draw it in a different color. So this is the first neuron, and this is the second neuron, and this is the synapse. Okay, so this is the synapse. So the neuron which is before the synapse, or the neuron which has the which which is the part of the synaptic terminal, it is known as presynaptic neuron, while there will be synapse here and then there will be next neuron. Okay, so this next neuron is known as postsynaptic neuron. Okay, I hope that's clear to you all. So let's say like this is the synapse and there will be a neuron which comes before, and then there will be a neuron which comes after. The one which comes before is known as presynaptic and the one which comes after is known as postsynaptic neuron. Okay, I hope that's clear to you all. So this presynaptic neuron will be giving out its output at the synapse, and then that output will be received as an input for the postsynaptic neuron. Okay, I hope that's clear to you all. So this is the basic structure of axon, and with this I'm done with the structural part of axon. I hope you the polarity part is also clear to you all. Okay, so okay, yeah, so this is the the first tracing, or the which was published of the action potential. So what happens is that neurons, they have the property of generating. So each cell has its each cell has certain potential. So let's just discuss this a bit. Okay, so let's say that this is the cell membrane. Okay, so the cell membrane, this is the inside of the cell and this is the outside of the cell. So let's say the outside of the cell is the extracellular matrix. Okay, and the inside of the cell, it is the cytosolic part. Okay, so this both of them are fluid, so now they will have certain ions. So there will be cations and anions in all, may it be extracellular matrix or the cytosol. So there will be certain, there will be certain concentration gradient. Let's say that the potassium is quite high here, and let's say the sodium is higher here. Okay, okay, then there is in the outside part there is chloride ions, and then inside part there are a lot of anions. A lot of anions besides chloride, there are a lot of different types of anions at the inside part. So here you can see the negative part is too much, like potassium ions plus the lot of anions, they are here. So compare when you compare to outside of the cell, the inside of cell has tend to have more of the negative charge, right? And that is why there will be negative charge on the inside of the cell. Okay, while outside the cell there are a lot of sodium ions. Sodium ion, there will be potassium ions as well. There will be here sodium ions and chloride ions, but the concentration is quite less. Okay, so we are not saying that they are absent here. The concentration is quite less. Even the potassium concentration is less, but the sodium concentration is quite high, and chloride concentration compared to the inside of cell is higher on the outside of the cell. So what happens is that basically the sodium ion concentration is quite high on the extracellular matrix, which gives overall positive charge compared to compared to the inside of cells. So this is all just remember that this is all comparative. Okay, so that is why there will be positive charge on the outside of the cell membrane. This is relative, this is not absolute. For example, like let's say that you are a good person, okay, and your friend is also a good person, but you both are good person, but you are you are too much, like you are much more better than him. So let's say that the goodness in you is more than him. So this is like relative. Okay, so both of you are good people, but he is you are better than him. So it's like it's relative. When you are more kind, you are more considerate, it makes you more positive, right? Compared to your friend, he is also a positive, but compared to him you are much more positive. So when we are going to do the comparison, you will be the positive, you will be the on the positive side, you will be the positive charge. So you will be the outside of the cell, let's say, like you are on the positive side. So compared to you, he is negative. Okay, absolutely he's not negative, he's a good person, but compared to you he's on the negative side. I hope I'm able to explain that in a better manner. So it's all relative, it's not absolute. So compared to outside, compared to extracellular matrix, the inner side of the membrane of the neuron. So here we were talking about neurons. Okay, inner side of the neurons, it is, it is negative in charge. Okay, so outside is positive while inside is negative. So this is the scenario in the resting state. So when the neurons are not receiving any kind of potential, any kind of stimulus, this is the scenario. So the outside of will be positive and the inside of the cell membrane, that will be the negative. Okay, so what happened was, the idea that the electrical signals, they are passed down through the neurons as the signals, that came when Hodgkin and Huxley, they did an experiment on the squid. Okay, so squid has giant axon, and that is why the squids were selected for this particular experiment. So what did they do was, they inserted a glass capillary electrode which had seawater in it, in the membrane of the, squid. So let's say that this is the squid membrane. Okay, so they inserted glass capillary electrode, and this has salt water. Okay, let me take. So this is the squid axonal membrane. Okay, so I'm just making a rough schematic. So they introduced a glass electrode, glass capillary electrode which had salt water. So this is salt water, and this is the, this is the glass capillary. So this glass capillary, when it pinches the axonal membrane, it is actually separating this part. So it is actually separating this part from the other part, right? So when it separates this part, and when you put an electrode, so you can specifically measure the, you can specifically measure this the membrane potential of that part. And when a stimulus arises, you can actually record the changes in the membrane potential. And this gave the idea that the electrical signals, they are passed down through the neurons. And neurons, they conduct the impulses, and that is the, that is the signal transmission in neurons. So for example, like there could be chemical signals, right? So chemical signals like neurotransmitter, but there are electrical signals as well in the neurons. And that idea was, it was established when this electrical impulses recording was done by Huxley and Hodgkin. So here you can see that the this was the resting membrane potential when there was no stimulus, and then suddenly the potential increases, and then again it goes back to the what it was before. So this is what we call as action potential, the change in the potential of the membrane, which is going to lead to certain action. This is the layman definition of the action potential. We will be discussing that in very detail in the upcoming lectures. But yeah, so this is how they establish the fact that the nerve impulses are conducted in the neurons. The electrical impulses are conducted in the neurons. And here you can see, this shows that they are, they are produced at certain distance. So actually, this axon, this is covered by the myelin sheath. So this is covered by the myelin sheath. Okay, myelin sheath. So around this is a myelin sheath. Okay. So this is myelin sheath. So myelin sheath, it acts as an insulation. It acts like an insulating part. It do not let the electron saying electron, it do not let ions leak through the membrane. Okay, so it provides the protection, it provides insulation, and due to the myelin sheath, there will be the fast transmission of electrical signals. So what happens is that there will be a gap, where there will be no myelin sheath, and this gap is known as nodes of Ranvier. Nodes of Ranvier. Okay, so this nodes of Ranvier, this part is very important, because at this part, the axon, at axon hillock, the action potential is generated, and it is regenerated at nodes of Ranvier, because nodes of Ranvier, it has specific composition of receptors, specific composition of ion channels, which are important for action potential regeneration. So the first action potential is generated at axon hillock, and then it will be regenerated at the nodes of Ranvier. And that is how the myelin sheath, it is, we will also discuss about this in conductivity lectures, like how the myelin sheath results into the fast transmission or the fast conduction of the nerve impulse. So I just forgot to discuss this part, that there will be myelin sheath which will be wrapping the axon, and at certain points where it doesn't wrap the axon, it is known as nodes of Ranvier. Okay, I hope that's clear to you all. So this is it about the neurons part. And in the last lecture I forgot to mention the two important principles given by Ramanukayal. Okay, so he already told us that there is the connection of distinct cells in the neurons. So he meticulously he meticulously observed a lot of different specimens, and he drew a lot of pictures which made him conclude those two principles. So the first principle was the principle of dynamic polarization, which states that the conduction of the electrical signals. So the electrical signals flow in one direction in nerve cell. Okay, so they do not flow randomly in any direction. There will be one single direction which will be followed for the nerve impulse conduction. So that is what is known as principle of dynamic polarization. I have already explained this part to you, like how from the dendrites are the input part, and how from the dendrite the nerve impulse, the signal is transmitted from dendrite to the axon hillock, then to axon, and then to the synaptic terminal. So we have already discussed this. So this was the very first principle which was given by Ramanukayal just by observing all of the specimens. So he did a really good job with observing those specimens, and he actually 130 years ago he drew all of the connection. He observed it, he did a lot of thinking part, and then he was able to come up with those principles. The second principle is the principle of connectional specificity, which says that the connections which are formed by neurons, they are not random, but they are specific. Okay, how are they specific? So yeah, we will learn about this in reflex action and all. But yeah, so the principle of connectional specificity says that nerve cells do not connect randomly, but each cell, rather each cell makes a specific connection. Okay, so like only the synaptic terminals of the presynaptic neuron. So it says that only the, here I already told you that only the synaptic terminals of presynaptic neurons, they are going to form connection with the dendrites of the next neuron, the postsynaptic neuron. So this is very specific. This is not random. So you can't expect the synaptic terminals to go and form connection with cell body. It is not normal. It is abnormal thing. So it is very specific. The connection formation is not random, it is specific. Only the synaptic terminal of presynaptic neuron is going to form connection with the dendrites of the postsynaptic neuron. So we already discussed both of those principles, but those principles were given by Ramanukayal. So the principles are the principle of dynamic polarization, which states that the electrical signal flows in one direction, and the second principle was that principle of connectional specificity, which states that those connection, this meticulous formation of connection by the nerve cells, that is not random, rather it is very specific. Okay, so with this we are done with today's lecture. In next lecture we will learn about different types of neurons. So we have just now studied all of the structural all of the structural parts of the neuron. In next lecture we will learn about different types of neurons. So this categorization will be based on different different properties, like the functional properties. It will be based on the structural properties. It will be based on the conductive properties of the neurons. So we will be learning about the types of neurons in the next lecture. Till then, see you. Bye bye.